Voice of the Sea, learning from experts across the ocean.
Welcome to Voice of the Sea. In this episode, we're in Guam and Palau, looking at poisonous marine organisms and their connection to cancer research. In Palau, scientists are collecting algae, corals, and sponges to screen them for cancer treatment potential. First, we're in Guam, collecting cone snail venom, which is leading to breakthroughs in cancer research.
Marine biologist Jason Biggs is studying cone snails and their venoms, which have many potential medicinal applications. Most people have only seen empty cone snail shells, but the animals themselves are fascinating marine predators. Jason talks about their biology and how to avoid being bitten by these toxic beauties.
So you've been a professor at the University of Guam since 2008?
2008. My work revolves around the pharmaceutical application of cone snail venoms for medicine, including certain cancer models, as well as malaria and other disease states. There are a bunch of different cone snails here. In fact, we probably have some of the most rare cone snails known, so we definitely have a lot of cool stuff to work on.
And what makes cone snail venom in particular suitable for this question of can it help fight cancer?
Well, we know that the cone snail venoms are very characterized to the point where we know a lot of the exact neurotoxins and we know most of the neurotoxins and where they act. So we know the receptors, and most of those receptors are on the nerves or part of the nerves. The interesting thing is a lot of the late-stage cancer phenotypes seem to express these same receptors, except they're in the body. For example, cone snails, because they're peptides, can't cross the blood-brain barrier. So, in essence, if you injected a cone snail venom into your system, it would work only on the body, the peripheral body. It wouldn't work on the brain. Well, cancer cells express these receptors that are only found in the brain. So if you, in essence, purify that specific toxin, you target something that's not inside of the body normally, except in this cancer cell. So you can home in on a cancer cell directly by attacking these late-stage cancer phenotypes.
How do you collect them?
We go out on the reef flats. You can go, it's a little bit easier at night because they come out. But even during the daytime, you can see them buried in the sand partially, and if you know the shape, the reason why they call them cone snails is because they're shaped like a cone. So if you get used to searching for that shape on the reef, it's really quite easy to pick them out. And you'd be really surprised at how many cone snails are actually on the reef where you don't even notice it.
I'd always thought that maybe incorrectly that they hid in holes.
They can bury under the sand during the daytime, and many do, but like other snails at low tide, they get a little frisky and start moving around in the sand. So you can see their trails right after low tide, and you can find them from their trails. Or sometimes they're parked and they didn't bury completely under the sand. And like I said, at night they come out, so you can find them just roaming around. Just separating out a little bit of the venom so that we can test it against some bacterial cells.
How did you collect it?
We try and milk the cone snails by faking them out and making them think that they're actually getting a meal or hunting the fish or snails that we put in front of them. But instead we actually have specialized tubes like these where the top is changed so that there's a piece of the prey, and they sting that instead of stinging the fish and puncture through the membrane that we have there and inject the venom into the tube so we can collect it directly that way. And these are just ultra accurate measuring devices where we can pick up exact volumes of different liquids depending on the size of this pipetman here. So here we can pick up something that's less than a drop of water. Here we can pick up something that's about a drop of water. And here we can pick up all the way to 1 milliliter. So we have to measure out the actual volumes in order to dilute out the concentrations that we're testing. And to do so, we have to do what's called a dose response curve, which means that we take and mix the same venom with the same number of cancer cells. The lower doses usually don't kill the cancer cells, whereas the higher doses do. So you try and figure out what level or what dose is enough to kill. So in essence, these wells become replicates in our experimental design. So then we just take the venom and measure out different quantities.
So each tube in that row is going to get the same quantity of venom?
Yes. And we usually do it in replicates of 4 to 5. So then after 24 hours of letting the cancer cells grow.
And would you just grow them at room temperature?
Usually it's in an incubator like this where it's at 37 degrees Celsius or body temperature. So this machine is a spectrophotometer. It can use different wavelengths of light to measure and count the cells in each of the wells. These are usually to measure cells, to count cells, or to measure different concentrations of proteins or nucleic acids inside of solutions. So here you can see it's just going through and reading the light that passes through each of the wells. You can see that some have higher light passing through and get a higher number, whereas some have much less light passing through. So this constitutes the less cells alive, more cells alive. Each cone snail species has a completely unique venom from another one. So although they work on the same receptors, they are slightly different, if not quite different from each other in terms of the actual venom component that they use.
And there's 200 or so different proteins that make up the venom for a particular type of cone snail?
Yes, each cone snail is estimated to produce and inject about 150 to 200 different active constituents in its venom. And with 700 different known species, we think there's at least 200,000 different active peptides to be characterized out in the cone snail venom.
So that's enough to keep you busy for quite a
For
while.
many careers can be made through working with cone snail venoms, definitely.
Have you ever been stung?
I have not been stung. You watch them very carefully if you pick them up, very carefully. Unfortunately, only one drug has been able to make it to the market so far. Omega conotoxin M7A, which is a conotoxin that's found in Conus magus, and it's directly derived from the venom itself. So it's the first and only FDA-approved drug that comes from a marine resource right now, but soon there are others that are going to follow in its footsteps.
Welcome back to Voice of the Sea.
In the venomous world, it's really interesting because there are many different strategies that the venoms attack. A lot of snakes attack the red blood cells and the actual blood system, the circulatory system of the prey. And that, in essence, coagulates all of the blood inside of the prey, and that's how the prey dies. Others, though, like spiders, often target the nervous systems of their prey, like cone snails do. Bees tend to target broad-acting cell kind of peptides that make the cells explode themselves. So there's a lot of pain, there's a lot of inflammation at the site.
But it's localized.
And it's localized. Cone snails, from what we've seen so far and from some of the work that I've done, show venom constituents of every other venomous animal. They attack the nervous system, they attack cells, and they attack the blood. Although instead of coagulating the blood, they tend to try and digest the blood or open up the wound more so that the venom can perfuse throughout the tissues of the body. The first set of studies on cone snail venoms were actually to find out what the poisonous constituents in the venom were, because the first interest that people had with them were collecting the shells themselves. And
They're very beautiful.
yes, they've been used for over 4,000 years as in ancient artifacts for jewelry and the like, because of that beauty. It just so happened that in the 70s they started trying to figure out why certain cone snails, if collected, would sting a human and be able to kill that human. And in fact, one of those cone snails, Conus geographus, exists on Guam, and there is one documented case where a military person collected Conus geographus and put it in their wetsuit and actually died 4 hours later from the sting. So that cone snail is actually still in the Naval Hospital in a jar of
That
ethanol.
individual.
That exact cone snail is still sitting there. In our lab, if we're going to handle the snail, we're required by the CDC to use Kevlar gloves. So we handle them with Kevlar gloves, and we always handle them by the back end of the snail. A lot of people in the islands have been told to hold a cone snail by the back of its shell. But from a lot of the videotaping that I've done so far, we can prove to you very quickly that the proboscis, or the stinging part of the shell, can reach well beyond the snail itself.
Back.
Yeah, so there's really no use in holding it in the back, other than it gives you a little bit more time to notice that you're going to get stung before the proboscis makes it to your hand. Usually immediately upon touching them, they go into their shell, and so you have a few seconds to move them around without worry of getting stung. It's usually only when you're sitting there talking with them or something, you know, or you lose track of what you're doing, that the danger starts to rear its ugly head. So.
The University of Hawaii Sea Grant College Program, helping coastal communities of Hawaii and the Pacific through research, education, and outreach. Serving the community from elementary to graduate students. Hawaii Sea Grant.
Welcome back to Voice of the Sea. Next, we're in Palau, talking with Pat Colin of the Coral Reef Research Foundation. Pat shows us his collection of algae, corals, and sponges, whose chemicals are being looked at as potential cancer treatments.
And what we do, we're contracted to go out and collect samples of marine invertebrates and marine plants, which is essentially algae, and then freeze samples of those, ship them off to the NCI laboratories in Frederick, Maryland, and then we identify and document all the samples. The samples are identified, the species, and with full collecting data. Marine organisms are very have a very high potential for drug development, much more so, interestingly enough, than, say, rainforest plants.
Why is that?
You could think of it this way: that you have this taxonomic diversity of invertebrates, from literally the sponges to the ascidians, which has developed over hundreds, if not billions of years. And then you have land plants, which have been around a long time, but the evolution across the entire spectrum of land plants is not as great as, say, even in just sponges. So that evolutionarily thinking, you've had this much broader spectrum of life that's had a chance to evolve interesting compounds. So that may be one reason. Terrestrial plants have basically, for anti-cancer compounds, have been a dry hole. There isn't a single anti-cancer drug developed from a terrestrial like rainforest plant. Maybe the compounds might be usable for something else,
Sure.
Antibacterial, antifungal, but for anti-cancer drugs, the rainforest has been a wash. That doesn't mean we shouldn't save the rainforest, but the idea that that the rainforests are going to cure cancer has pretty well been disproven, unless something changes remarkably. I like to say the kind of things we're interested in are what you would scrape off the bottom of your boat when you haul it out, the fouling organisms. Falling organisms are great for us.
Do you ever collect from the bottom
Oh,
of boats?
absolutely. Bottoms of boats have been one of the best sources of this stuff. We see a derelict boat sitting where, to us, it looks like a gold mine. A gold mine in the sense that we can collect a lot of samples, not make a lot of money, because nobody really makes money doing this. But it's like, there's all these samples, piers, docks, pilings, any kind of structure, because it presents a different environment. So what might be really common around the pier is not what grows on the pilings.
Sure.
That's a whole different environment. So you can get what's on the bottom, and then you get what's on the pilings. And we're continuing to do this work for the NCI. This is sort of our home base. We've actually worked in about 22 different countries worldwide over the years.
Wow.
But Palau has been a major focus of the work. Our job as the contractor at the NCI is to collect, identify, and document the marine invertebrates and marine plants that we're collecting for anti-cancer drug screening. So that the actual sample that's used for testing, we try and get around 1 kilo wet weight of whatever it is we're going after. So obviously if you want 1 kilo, which for the non-metric is 2.2 pounds, that's a fair amount of
quite a
material.
bit.
It's what you would basically hold in your hands like this. And if it looks like there's some compound in there that has good anti-cancer activity, they can actually do most of the chemical structure work, so they can purify it, do the structure, and identify the compound, all based on 1 kilo. The idea is to collect enough, and these are very common things. And what we're doing really has no impact on the environment. It's things that you'll see vast fields of on the bottom, and we take a sample, and literally, if you went there 5 minutes later, you would never know that the thing was even collected. So that's what we're looking for, and hopefully the entire process, one sample, and we end up someday with a cancer drug. There are samples that were collected in the 1980s that are just now being taken into the stage of actually becoming a drug.
And that might be one of the reasons why it's important to collect that whole sample
Yes.
in the beginning.
Yeah. And sometimes, things come and go. We see this all the time in shallow water marine environments. You'll have a bloom of some particular species that you've never seen before, and then a year or two later, it's pretty well gone. It's somewhere, but you don't know where it is anymore. So that we can take an opportunity, advantage of opportunities where something has a bloom and go out and collect it.
How are your samples in here organized?
Well, they're all organized. They're by phyla. Let me just grab one here. Every sample has a number, and it has a sample. This is preserved in either alcohol or formaldehyde. Most things are in alcohol. A few other groups have to go into formalin or formaldehyde. So it has a unique number. This number is used for all the testing by the NCI. We have it in a database, electronic database with photographs. We photograph everything extensively underwater. Now, many of these things are actually undescribed species. They've never been given a scientific name or described. So for us as biologists, we're not chemists, we're biologists. One of the most satisfying things is that our taxonomists then are describing many of these things as new species. So we've had around 200 new species described from the material we've collected in the last 20 years. But there's a wall of sponges over here. There are probably 500 new species sitting there. There aren't enough taxonomists to do them all. These are examples of the types of things we collect. We collect 1 kilo wet weight sample, we freeze it. We have to ship it frozen from here, Palau, or wherever we are, all the way to Frederick, Maryland, which is an interesting exercise at times. We have insulated boxes. We use dry ice if it's available. So we have always gotten them to Frederick frozen. And then those samples, the raw sample is taken and is ground up. Actually, they use a hamburger grinder most of the times. the sample is ground up into flakes or small bits, and then it's put into a column like you see here, a glass column. This has a stopcock, and then it has a flask below. So first they put water in here, okay, so that any compound in this material that dissolves in water will dissolve into that water. Not everything, not the total amount, but most of it dissolves into the water. Then it's drained into this flask, and then the water is extracted by freeze drying. So think of it as freeze-dried coffee, where you don't cook it. This could be coffee beans, and coffee is just an extract of coffee beans. How they do the testing is kind of an interesting story. You can culture human cancer cells, tumor cells, in a laboratory, basically, in test tubes, what they call a cell culture. They take those living human tumor cell types, and they actually screen against about 60 different cell types, tumor types. They take a little plate which has 96 little holes in it, 96 wells, and they put tumor cells in those. They have a few other controls. And to each one of those samples of the tumor cells, they add a little bit of this raw extract from these. And the idea is that, okay, if something in that extract kills the cancer cells, okay, there's something that's cytotoxic. It has potential anti-cancer activity. That's great, but the problem is that something like 30% of these samples will kill cancer cells in a test tube, basically.
Sure.
However, most of these things are so toxic, they kill everything.
Right.
So you'd kill the patient treating the cancer. So the idea is that you have to find a sample which has a differential activity, say something that stops cell division. Of course, cancer cells are dividing very fast, so they really like things that have an effect on cell division and can stop it. Instead of just poisoning the cell outright, if it can stop the cell division, then okay, this is a good prospect. It's generally estimated that only about 1 in 10,000 samples will ever be usable as a drug, and it may actually never even be developed as a drug. It may not be better than what's already available. So, we have to collect, the idea is to collect as broad a spectrum as possible, hoping that you just find that one that is the magic bullet. We haven't found it yet, but it may be out there somewhere.
Exploring Our Fluid Earth is a dynamic curriculum developed by the University of Hawaii's Curriculum Research and Development Group, teaching ocean science concepts through the disciplines of physics, chemistry, biology, and ecology. Exploring Our Fluid Earth is now available freely online. Find out more at exploringourfluidearth.org.
The Curriculum Research and Development Group in the College of Education at the University of Hawaii at Manoa. CRDG has been providing quality educational programs and services for over 40 years, serving students, teachers, parents, educators, and experts around the world and here in Hawaii. The Curriculum Research and Development Group, improving schools, improving education. CRDG.
COSI Island Earth is working to establish new avenues for connecting research scientists with educators and communities. COSI Island Earth is working to enhance the science and ocean literacy of our island residents and visitors. COSI Island Earth is working to connect scientific research, traditional knowledge, and ocean policy. COSI Island Earth, bringing together university, government, research, and community partners to enhance ocean literacy and engage all ocean users in stewardship.